Evolution is the gradual process by which species of organisms undergo changes over time due to variations in their genetic makeup. This natural mechanism drives the development of life, allowing organisms to evolve from simpler forms into more complex and diverse beings. Through evolution, traits that enhance survival and reproduction become more common in populations, shaping the diversity of life on Earth. Among scientists, there are different schools of thought regarding the origin and development of life. Two prominent perspectives are special creation and organic evolution. This view, associated with scientists like Carl Linnaeus, holds that all species were created in their present form by a divine being. According to this perspective, life forms were individually designed and have remained essentially unchanged since their creation. Then we have the organic evolution one, advocated by scientists such as Jean-Baptiste Lamarck and Charles Darwin. It argues that life forms have evolved over time through natural processes. This school of thought emphasizes that species change gradually due to genetic variations, natural selection, and other evolutionary mechanisms, leading to the diversity of life observed today. About 3.5 billion years ago, life is believed to have begun possibly near hydrothermal vents on the ocean floor. These vents are like cracks in the Earth's crust that release superhot, mineral-rich water. According to the hydrothermal vent hypothesis, these vents provided the perfect environment for life's building blocks to form. Inorganic molecules like hydrogen, carbon dioxide, and sulfur mixed together in intense heat and pressure, creating simple organic molecules such as amino acids. Over time, these organic molecules started interacting and combining in more complex ways, eventually forming the first living cells. The earliest forms of life were prokaryotes, specifically a group called Archaebacteria. These primitive organisms were heterotrophs, meaning they relied on organic compounds for nutrition. They likely fed on the organic molecules that were formed through the interactions of inorganic compounds in their environment. After Archaebacteria, the next major step in the evolution of life was the emergence of photosynthetic bacteria known as cyanobacteria. Unlike their predecessors, these bacteria were autotrophs, meaning they could produce their own food through photosynthesis. Cyanobacteria use sunlight to convert carbon dioxide and water into organic compounds, releasing oxygen as a byproduct. This process not only provided a new source of energy, but also began to increase the oxygen levels in the Earth's atmosphere, paving the way for the evolution of more complex life forms. As prokaryotes began evolving into eukaryotic cells, two key theories explain how this transformation happened. First, we have the membrane invagination theory, which suggests that the membrane of an ancestral prokaryotic cell folded inward, creating internal compartments. This initial invagination enclosed the cell's genetic material, forming a nucleus, which is the defining feature of eukaryotic cells. As the membrane continued to fold inward, it formed additional pockets which evolved into various organelles with specialized functions, such as the endoplasmic reticulum for protein and lipid synthesis, the Golgi apparatus for processing and packaging molecules, and other organelles like mitochondria and chloroplasts. This process allowed cells to develop the complex internal structure characteristic of eukaryotic cells. The endosymbiosis theory offers another explanation for the evolution of eukaryotic cells. It suggests that a large ancestral prokaryotic cell engulfed smaller bacteria, but instead of digesting them, the host cell retained these bacteria as internal symbionts. For example, consider aerobic bacteria that use oxygen to produce energy in the form of ADP through aerobic respiration. Now, when a large prokaryotic cell engulfed such aerobic bacteria, these bacteria evolved into mitochondria, The organelles, known as the powerhouses of the cell due to their ability to generate large amounts of ADP. Similarly, when a large prokaryotic cell engulfed cyanobacteria, which are organisms capable of photosynthesis and producing their own food, the cyanobacteria were retained and eventually evolved into chloroplasts. This gave the host cell the ability to perform photosynthesis, allowing it to become autotrophic. The evidences supporting the endosymbiosis theory includes the fact that both the stroma of chloroplast and mitochondrial matrix contain their own circular DNA, which is similar to the DNA found in bacteria or prokaryotes. Another interesting thing is that both chloroplasts and mitochondria have 70S ribosomes, the same type found in prokaryotes, rather than the 80S ribosomes which is typical of eukaryotic cells. Also, these organelles replicate independently of the cell through a process similar to binary fission, just like prokaryotes do. This independent replication is unlike the way other organelles within the eukaryotic cell divide. These similarities provide strong evidence that mitochondria and chloroplasts originated from symbiotic relationships between the ancestral prokaryotic cells and larger host cells, eventually evolving into the organelles we see today in eukaryotic cells. In the next video, we will discuss the theories of organic evolution. We will explore pre-Darwinism, the foundational ideas of Darwinism, and how these evolved into New Darwinism. Thanks for watching and see you next time.